Yeah, okay. So it's my great pleasure and the great pleasure of Michel Monje to welcome all of you to the third Cancer Neuroscience webinar of the Brain Prize. And this third Brain Prize webinar will now lead us to, well, still a little bit outside the brain. So our body is full of nerves and full of the peripheral nervous system and also other neural features driving cancer. And this is an aspect of cancer neuroscience that is probably not so aware to many neuroscientists. So today we have three really wonderful and speakers who have really have really contributed in a very significant way to the advance of cancer neuroscience outside the brain. And we are really looking forward to your talks. And I hand over to Michel to introduce the first speaker.
Wonderful. Well, it's really a pleasure to introduce Timothy Wang, who is truly a pioneer in investigating how peripheral nerves interact with cancer. He is the Dorothy L. and Daniel H. Silberberg Professor of Medicine and Vice Chair for Academic Programs in the Department of Medicine at Columbia University Irving Medical Center. He's also co-leader of the program in tumor biology and microenvironment for the Irving Cancer Center. A graduate of the Vagelos College of Physicians and Surgeons at Columbia University, Dr. Wang trained in gastroenterology at Massachusetts General Hospital, Harvard Medical School, and then joined faculty there, returning after that to Columbia in 2004 as division chief in digestive and liver diseases. His laboratory studies gastrointestinal cancers, gastric, esophageal, pancreatic, and colon cancers, and the role of the tumor microenvironment in those diseases. He investigates both early and late stages of GI cancers and the role of interactions between progenitor cells and stromal cells, with a particular focus on nerve cancer interactions. An early investigator in the cancer neuroscience field, he studied both sensory and autonomic nerves, developed a model whereby cancer nerve connections generate a larger neural circuit that leads to changes in the TME, with the goal of bringing new therapies to patients at the bedside. Thank you so much for joining us, Dr. Wang, and we look forward to your talk.
Okay, so let me just share. Okay, hope you can see this. Yes, so it's a pleasure to be able to give this webinar talk in honor of our Brain Prize awardees, Drs. Michel Monje and Frank Winkler. Congrats to our awardees, and many thanks again to our organizers from the Lundbeck Foundation. So as mentioned, I'll be speaking about our work on peripheral nerves, and in particular on the role of sensory nerves, which through bidirectional interactions directly promote stomach cancer. I'll describe our published work and at the end mention some of our more recent directions. Okay, so there's now increasing evidence that peripheral nerves, both autonomic nerves and sensory nerves, are increased in solid tumors and play a central role in their pathogenesis and progression. And we've now, as we've been hearing in the webinar so far, cancer neuroscience is now a well-established field, and neural regulation of solid tumors is increasingly viewed as a possible therapeutic target. But in particular, I'm going to focus on sensory nerves, which have been a recent focus in the field. And I thought I'd start off by listing some of the unanswered questions in the field that we and others have been trying to address. How are these nerve cancer interactions initiated? What types of nerves are most increased in the tumor microenvironment? Do nerves act only in paracrine fashion? Whereas we'll be hearing in this and other webinars, are there direct electrical connections? Do cancer cells signal to nerves to create this electrical neural circuit? And how do the neural signals actually promote cancer growth and or invasion of spread? And are nerves then a viable therapeutic target? I'll be speaking mostly in this particular talk on gastric adenocarcinoma, which is now less common in the United States, but worldwide remains the 5th most common cause of cancer-related mortality, responsible for nearly 800,000 deaths every year. It is one of the recalcitrant cancers, with a poor survival rate here listed at 31% over 5 years. And Helicobacter pylori infection is known to be the major risk factor. Nevertheless, rates are rising in the US, particularly in patients under the age of 40, many of whom do not have H. pylori infection. Several decades ago, we noticed that gastric cancer tended to rise most commonly on the lesser curvature of the stomach and at the antral corpus junction. And this is an area that was known to be rich in neural innervation, largely supplied by the vagal nerve. So we began to ask questions about the role of the vagus nerve in gastric cancer. This slide now summarizes a lot of work from the past 10 years on the effects of vagotomy in suppressing gastric tumorigenesis. About a decade or so ago, we carried out surgical vagotomy, both unilateral and bilateral vagotomy, in 3 separate gastric cancer mouse models, and showed that vagal denervation suppressed cancer growth, but only on the denervated side with unilateral vagotomy, indicating a very direct effect. We confirmed that SHAT-positive cholinergic nerves were increased in gastric cancer models due largely to the upregulation of a growth factor, nerve growth factor, and that cholinergic nerves then released acetylcholine to activate the Wnt-Hippo pathway through the muscarinic 3 receptor. Now, at the time of this study, which we carried out around 2013, we were focused largely on cholinergic nerves, not really thinking much about sensory nerves. But on later reflection, sensory nerves were an obvious candidate. And that is because vagotomy also reduces the abundant vagal sensory nerves that are present within the vagus nerve. And it was known that NGF in particular promotes the outgrowth and survival mainly of sensory nerves, and that in the GI tract, as listed here in these references, sensory nerves were known to regulate a number of secretory cell types. Finally, in the last 10 years, a number of studies have demonstrated roles for sensory nerves in solid tumors. Thus, we decided to investigate the role of sensory nerves in gastric cancer, asking the question as to whether they were in fact direct bidirectional interactions with electrical and chemical communications similar to the land-breaking work in gliomas. Now, we first started off looking at the neural expansion in gastric cancer and the changes in a variety of different gastric cancer mouse models. This bar graph on the top right shows the density of various nerve types. And as you can see, at baseline, cholinergic VCHAT-positive nerves were the most abundant. But within the cancer models, you can see that the greatest increase in most abundant nerves were the sensory nerves positive for CGRP, CGRP standing for a neuropeptide calcitonin gene-related peptide. And we confirm this, as shown in the bottom right, that immunostaining of gastric cancer tissues, gastric cancer microarrays, showed marked upregulation of CGRP sensory nerves. Interestingly, while in our syngeneic mouse models with the ACKP gastric cancer cell line, when implanted orthotopically, it showed abundant increase in peptidergic CGRP-positive sensory nerves. When these same tumors were implanted subcutaneously, they showed a predominance of non-peptidergic nerves, NF200 piezo2 positive neurons, indicating that severe limitations of these subcutaneous cancer models. Now, to address once again the mechanism for the marked expansion of nerves in gastric cancer, we examined by qPCR the expression of neurotrophins in purified gastric cancer cells across these multiple gastric cancer models, and found again that NGF, nerve growth factor, was by far the most upregulated neurotrophin. In situ hybridization showed that NGF was expressed at baseline in rare epithelial cells in the gastric isthmus or progenitor zone, shown here in this top right panel, but was consistently upregulated following gastritis, peptic ulcer disease, any kind of injury to the stomach, and in particular in cancer, it was markedly upregulated in this sort of progenitor zone of the stomach. For example, up to KRAS activation or carcinogen exposure, NGF was clearly upregulated, as shown in the bottom immune staining panel. Overall, we found the increased NGF immunostaining in the gastric glands in all of our gastric cancer mouse models, as shown in the top right. And NGF upregulation consistently led to an increase, a marked increase in the density of CGRP-positive sensory nerves. And this increase in sensory nerves could be blocked using the NGF TRAC receptor inhibitor and entrectinib. Thus, these data strongly link NGF to the expansion of CGRP positive sensory nerves. Now, in order to look at, again, the origin of these sensory nerves within the stomach, we carried out detailed retrograde tracing using a variety of different retrograde tracers, as shown here. This included FAST Blue, CTP Alexa Fluor, AAV HSyn EGFP, and these were injected into the anterior or posterior wall of the stomach. And we then looked for these tracers in the nodose ganglia, in the mouse called the JNC, or the dorsal root ganglia. And we found consistent expression of each of these tracers in the ipsilateral nodose or DRG ganglia. And the DRGs, they range from T7 to T13, as shown here in the top right. And immunostaining of these sensory nerves showed that the peptidergic CGRP positive, or the non-peptidergic that were labeled with isolectin B4 nerves, are mutually exclusive types, but the vast majority, 80 to 100%, were CGRP positive. Now, to trace the gastric cancer connections specifically, we used the Delta G rabies deficient virus expressing DS-RED and sorted the trace neurons from the neurons of the nodose ganglia and DRG, and all of the tracer labeled neurons expressed CGRP, and they actually showed higher levels of the NGF receptor TRAC-A by flow sorting. Now, the receptor for CGRP consists of a heterodimer between the calcitonin-like receptor, CAL-CRL, and RAMP1. And we found on single-cell RNA plots of both mouse and human stomach that both were expressed, but that in cancer, it was RAMP1 that was specifically upregulated, confirmed by immunostaining and in situ hybridization. Now, as shown here, in situ hybridization for RAMP1 in the normal stomach showed expression both in the stem or progenitor zone here at the top of the glands, and also in the bottom of the glands in the secretory cell types, sheath cells. But in the cancer models, such as this triple transgenic cancer model shown here in the immunostaining panel on the right, you can see a marked expansion of RAMP1 in most or all of the dysplastic cells, showing that this could in fact be a part of the pathway driving the expansion of these dysplastic cells. Bulk RNA seeking of human tumors, shown here in the top right, showed a strong correlation between RAMP1 expression in gastric cancer and poor survival, as shown here in this curve, shown in red. Now, as shown in the single cell plot in the bottom left, RAMP1 co-localized with a subpopulation of progenitor cells that were strongly expressing Ki67, and these were increased in the stem cell zone in our cancer models. Now, we postulated that CGRP might regulate gastric stem cells, normal stem cells, and this was confirmed, as shown in the bottom, in our 3D organoid models, where the addition of CGRP increased the growth of gastric organoids, indicating a clear effect on gastric stem cells. Now, to further study this relationship between CGRP-positive sensory neurons and the proliferative epithelial cells in the stomach, we utilized a model of chemogenetic activation of sensory neurons. And we crossed TRPV1 cream mice that targets nociceptive neurons with the ROSE26 HM3DQ line, expressing this modified version of the human muscarinic 3 receptor, a DREAD or designed receptor that binds specifically to a DREAD agonist such as clozapine N-oxide or CNO. Mice were given CNO for 40 weeks, and as shown in the top left, they developed marked gastric hyperplasia with a twofold increase in the proliferative zone of Ki67 positive cells. We also gave CNO to mice following a gastric ulcer model, and as shown in the top middle here, again, this induced marked proliferation and a marked decrease in ulcer area by 10 days, indicating faster gastric ulcer healing. The sensory nerves normally play a clear role in mediating regenerative healing responses, and this is likely the sort of pathway that it has been usurped by tumors. Now, chemogenetic activation of the nociceptive neurons in our models of gastric cancer, the MNU model, M-nitrosourea shown here in the bottom, a chemical carcinogen model, or our ACKP orthotopic cell line model shown here on the right. In both, this led to faster tumor growth, increased tumor area, and as shown in the bottom right, a markedly decreased survival. In both models, treatment with a CGRP inhibitor, rimegepant, a commonly used migraine drug, inhibited the effect of sensory neurons on tumor growth and led to prolonged survival in all of our tumor models. Now, to test whether sensory neurons are necessary for gastric cancer progression, we bladed them using genetic models such as TRPV1 queen mice crossed to Rosa 26 DTR mice, treating them with diphtheria toxin. DT treatment led to a significant decrease in CGRP neurons in both the DRG and the dorsal root ganglia and the nodus ganglia shown here in the top left. And it also inhibited tumor growth shown here on the right, with a significant decrease in tumor area in both the MNU model and the ACKP orthotopic model. We found similar results with injection of resiniferatoxin or RTX, an analog of capsaicin that is cytotoxic again to TRPV1 positive neurons, and which also ablated sensory nerves and inhibited the growth of both cancer models, as shown before. Now, the activation of sensory neurons might be mediated in part, might also play a role in gastric cancer migration, metastasis, and most of what I've shown you before has been the effect of sensory neurons in early tumor growth. And so we really wanted to address the role of sensory neurons in metastatic disease. We use three different approaches here. In the top left, we show the in vitro co-culture of nerves and cancer cells. On the bottom left, this is an analysis of portal vein of migration. And the top right, you can see the effect in this metastatic model. I'll just start with the in vitro models where we co-cultured our chemogenetic sensory nerves shown in red with cancer cells shown in yellow. Treatment with the dread agonist CNO led to activation of nerves, and as you can see, migration of the cancer cells from the right towards the sensory neurons, suggesting that in fact RAMP1 signaling actually induces migration of cancer cells. In our ACKP orthotopic model of gastric cancer, the tumor is planted in the stomach. Again, activation of these sensory neurons using this chemogenetic method led to a marked increase in circulating tumor cells, CTCs, isolated from the portal vein. Finally, on the right, we show our metastatic model where the tumor is implanted in the stomach, and the mice are then subjected 4 weeks later to gastrectomy and Roux-en-Y esophagojejunostomy. And normally in this model, the tumors do occasionally metastasize to the liver, shown in the top panel, with about 20% of the mice developing liver metastasis. With activation of the sensory nerves with CNO treatment, we saw a marked increase in metastasis, a 4-fold increase or more, with confirmed metastasis to both the lymph nodes and the liver, driven again by sensory nerves. Thus, sensory nerves can clearly promote metastatic disease along with primary growth of the tumors. Now, we're interested in the interaction between these sensory nerves and gastric cancer cells, and we examined this primarily through our in vitro 3D culture models, as shown here, where again we cultured DRG neurons, shown here in TD tomato red, in our mouse gastric cancer spheroid, shown in yellow, isolated from our triple transgenic mice. And when we carried out co-culture, shown here, they resulted in very close connections between the sensory nerve outgrowths, the axonal neurites, and the cancer cells. You can see the red neurons are actually touching the cancer cells. Now, when we treated the culture media with CNO, activating the sensory nerves, this led to greater migration of cancer cells towards the DRG, as well as increased outgrowth of the neurites, and much greater connections, as shown here, and particularly on the bottom, you can see the overlap of the nerve terminals with the cancer cells. Now, this could be blocked by rimegepant treatment, again, this CGRP antagonist, suggesting this interaction is driven by CGRP signaling. Now, interestingly, we isolated gastric cancer cells that showed these neuronal connections, as well as the DRGs that showed these neuronal connections, and compared them to ones that did not show connections. And when we looked at the gastric cancer cells, we showed upregulation of a number of transcription factors known to be involved in neuronal programming, such as NeuroD1 and ASCL1. In addition, the connected neurons showed this upregulation of synaptophysin, particularly at the connected sites, suggesting that, in fact, they were developing not true synapses, certainly, but these synapse-like connections, suggesting that they might actually be directly communicating in a very close way. Now, to look at how the gastric cancer cells might be communicating to the sensory nerves, we cultured our neurons, cancer cells, but in the case of the neurons, we infected them with viruses expressing a number of G protein-coupled activation-based neurotransmitter sensors, so so-called GRAB sensors, several dozen expressing a number of different known sensors for these sensory neurons. And then we co-cultured them with ACKP gastric cancer cells that expressed the channel rhodopsin receptor, activated them with blue light, and then determined what receptors really were activated by these ACKP cancer cells. And as we can see in this slide, there were two receptors, adenosine sensor and a glutamate sensor that both showed signals. You could see a little yellow signal within the nerve cell bodies, indicating they were in fact activated by glutamate and adenosine. And so this was a mechanism by which the cancer cells could actually be talking to these sensory nerves. Now, to look in more detail at whether the nerves in the cancer cells were forming some kind of neural circuit, we turned to calcium sensors. First, this JR-Gecko-1A DS-RED calcium sensor expressed within the gastric cancer spheroids. And on the right, later I'll tell you about the GCAB6 sensors expressed within the sensory nerves. So first, we carried out co-culture of the chemogenetically expressing sensory nerves with the cancer cells expressing this calcium sensor. We activated the sensory nerves with CNO and showed that within 1 to 10 minutes, you could see this red color forming in the top left here in response to chemogenetic activation of the sensory nerves, indicating that the gastric cancer cells were in fact showing this calcium flux, suggesting depolarization of the gastric cancer cells. And this could be blocked by rimegepant. On the right, you can see that we co-cultured this was a in vivo study where we implanted gastric cancer cells in the stomachs of mice expressing the channel rhodopsin receptor, activated the cancer cells with blue light, and then looked for changes within the ipsilateral nodus ganglia. And within 1 to 2 seconds, you could see the change in color, the appearance of the red color is shown here in response to signaling from the gastric cancer cell, indicating a clear signal from the gastric cancer cell to the sensory nerve, indicating the cytodirect connection. In response to signaling, we also saw a release of CGRP peptide in the tumor, suggesting the presence of this sort of neural circuit mediating the response. So how do sensory nerves through CRGP, through RAMP1 receptor, actually cause gastric cancer growth directly? We carried out this sort of bulk sequencing study of ACKP organoids cultured with or without nodus ganglia, or with or without activation of the ganglia using CNO, the chemogenetic activator, and carried out gene set enrichment analysis. And in every comparison, we group 2 versus group 1 and group 3 versus group 2, et cetera, we showed that the RB E2F pathway was activated in every single comparison. And we showed further through additional signaling studies that this could be mediated through PI3 kinase, AKT, or CAM kinase using a variety of blockers, and confirmed that this pathway correlated with CGRP staining using phospho-RB staining in human gastric cancer tissue microarrays. Thus, through activation of E2F and inactivation of the RB pathway, CGRP can mediate gastric cancer growth. So what I've shown you so far is that gastric cancer exhibits these synapse-like connections. Certainly not, the data is insufficient for true synapses, but we see the extremely close juxtaposition of cancer cells and nerves, upregulation of neuronal transcription factors in cells that are connected, upregulation of synaptophysin in the connected nerves, this bidirectional communication and functional electrical connection between gastric cancer and nerves. And so far to date, we've been unable to demonstrate true synapses through electron micrographs and single cell patch clamping, although we have some patch clamp evidence. But this is a fast moving field. And as you'll hear from others, and in particular, I'll just highlight a recent study on pancreatic cancer where they've sort of described these sort of pancreatic cancer pseudosynapses, and you'll hear more about lung cancer, but certainly in the case of lung cancer, they've been able to develop, certainly in the brain, evidence for true synapses between certainly small cell lung cancer and brain metastasis. So where is this field now sort of going? I guess our vision of this is that there is this sort of fairly direct electrical connection between sensory nerves and gastric cancer cells. And now looking beyond this into the tumor microenvironment, and certainly we have data that there is additional signaling to LEPR-positive fibroblastic cells within the gastric mucosa. And these cells express highly RAMP1, and we've been able to show that the sensory nerves also cause electrical changes within the fibroblasts, these CAFs, and that these CAFs probably play also a large role in promoting both growth of the cancer cells and the immune suppression. We also have data now that they're signaling to CD8-positive T cells of sensory nerves in the gastric cancer microenvironment, larger effects on tumor draining lymph nodes. And now we have evidence that there is data CNS signaling where the activation of these sensory nerves by gastric cancer cells lead to activation of specific centers in the brain that really is of interest to many people in the field. And finally, gastric cancer is preceded by the development of gastritis, atrophy, and metaplasia, pre-neoplastic states, and we begin to see changes in the neural circuits in these pre-neoplastic states that may be leading to heightened gastric cancer risk. So how does this affect therapeutic implications? We've been carrying out now studies looking at the combinations of CGRP receptor inhibitors with other standard cancer therapies in gastric cancer. The standard therapy in gastric cancer is a combination of chemotherapy, typically 5-FU and oxaliplatinum, in combination with an immune checkpoint inhibitor such as anti-PD-1. In our mouse models, these show a little bit of additive effects, but when you add in a CGRP inhibitor such as rimegepant, it suddenly becomes much more effective with extending survival probably close to 100 days at least. And this looks very promising in terms of bringing these sort of neuro-targeted therapies to the clinic eventually. So just to summarize then, we believe that gastric cancer cells really establish this sort of neural circuit with sensory nerves through NGF, and I'm sure other neurotrophic and axonal guidance pathways. We've just touched the surface of this. The cancer cells can actually signal directly to sensory nerves, to glutamate, adenosine, likely other pathways as well. And this leads to an activation of this neural circuit, leading to the release then of CGRP, possibly other neuropeptides that we have not yet investigated. But this neural circuit then can be extended more broadly, leading to interactions with immune cells, fibroblasts, possibly up to the brain, possibly establishing a neural circuit that could be long-lasting. And so I think this is an exciting area for the field. A lot of this has been sort of driven by this concept of electrical communications from the winners of the Brain Prize here, Drs. Michelle Monje and Frank Winkler, who really inspired us all to investigate these pathways further. So I'll just let's stop here and thank my laboratory, many of the members who've been carrying out these studies, and thank you very much for your kind attention. Thank you.
Thank you so much, Tim, for that beautiful talk. I think we have time for a few questions. Maybe I'll kick off with a question and then ask others here on the panel to comment. I was wondering that this enrichment in NeuroD1 and ASCL1 expression in the gastric cancer cells that are more connected or touching the sensory nerves more is really fascinating. And I was wondering if CGRP induced that phenotypic shift, if it induced the transcriptomic expression of these important transcription factors.
Yeah, that's a great question. I mean, we've done this isolating these cells in culture with co-culture with nerves, both DRG and nodus ganglia. But I guess we'd have to go back and look at our bulk RNA-seq data. But we could also do this with direct co-culture of these organoids with CGRP directly. I think that's what you're implying. Yeah. Whether or not it's CGRP or it could be something else secreted by the nerves that's mediating this. So that is a study that we really need to do. So thank you for suggesting that.
Yeah, no, it's really fascinating, and also fascinating as you begin to get into this whole body communication between the gastric cancer and brain changes, and how that is mediated by sensory afference is really fascinating. So excited to learn more about that.
Yeah,
Others
thank
on
you.
others on the panel.
Yes, Frank.
Filippo.
Or Philippe,
Yeah,
yeah.
great talk, Tim. Thank you so much. But Filippo will start, please, and then I'm second.
Yeah, thank you so much for that amazing talk. I wanted to ask in the same direction as Michelle, because a lot of the genes that you mentioned, ASHL1, NeuroD1, are high in synaptophasing, are really markers of small cell lung cancer, which I will talk about in a second. And small cell lung cancer always has a mutation of RB1, which you also see being involved in the proliferation. And interestingly, there's data now suggesting that under therapy, other types of cancer also differentiate in that direction if they lose RB1 and P53. So there seems to be some kind of genetic mechanism leading from RB1 loss or phosphorylation to the expression of these neuronal genes. Have you ever looked in that direction or thought about it?
Yeah, I thought about it. I mean, I guess in other tumors, and one of the things that seems to come up is a lot of these tumor suppressors, when they become inactivated, somehow lead to neuronal signaling. And that's been an under-investigated area, I think, in general. We had not specifically looked at that by directly looking at these genetic mutations ourselves, but I think it's a wonderful thing. I mean, people have been wondering about these tumor suppressor genes and first thinking that they're entirely cell autonomous effects. And I think there's now emerging data with a lot of them that, to some extent, many of them, when inactivated, lead to changes in the microenvironment. And so, but I think the one area that's not been investigated in P53 or RB has been neuronal connections. So I think to some extent, maybe we should look at that more carefully. And obviously these are genetic studies and it can be done quickly in cell lines and so on. But I think that's really a great suggestion for future studies. Thank you.
Thank you.
Yeah, thank you, Tim. A great talk. You mentioned that you are looking into the pre-cancerous stages of inflammation, etc. I think that's wonderful. And I know that others are looking in similar direction in, for example, lung cancer, and when it comes to tobacco and other particle, etc., and find interesting results. So my question is, can you share a little bit of these kind of first insights? And do you see Helicobacter pylori specific mechanisms of changing, for example, increasing sensory nerve density or something like that? Helicobacter pylori is kind of one of these germs that is well known to also be an important part of this blood. The gut-brain axis is known to influence the CNS, blood-brain barrier, etc., etc. So, is there anything specific to H. pylori, and what are your first insights here?
Yeah, no, that's a great question. Is it just specifically H. pylori? So we've been looking at different injury models, gastritis models, chemical, carcinogen, others. I mean, one of the in the GI field, there's this actually tamoxifen, which everyone uses for lineage tracing. At high doses, it induces damage to the stomach specifically, and it causes a loss of secretory cell types, similar to that seen with H. pylori. So we've been using that and other models, and ulcer models as well. And so when you lose some of these secretory cell types, you get a regenerative and an inflammatory response. And so one of the things, so that leads to changes in the sensory nerves. As I mentioned before, the sensory NGF gets upregulated in injury to the stomach. Sensory nerves come in and play a role in healing of the mucosa. And I think there's a similar story in the skin as well, that sensory nerves play a role in healing the skin injury. So what was surprising was that these changes in the sensory nerves are long lasting, and they get reprogrammed in the sensory ganglia in a way that's somewhat similar to what you see in cancer. So our question is, since there's no cancer cell, what is causing these sorts of changes in the sensory nerves? And it turns out, I think probably what goes on is that the inflammation, specific inflammatory cytokines, may be reprogramming these sensory nerves. And I think I saw a recent paper, I think in Nature elsewhere, that was looking at colitis, for example, and changes in sensory nerves that occur in colitis. And it's a similar sort of story. So that is, I think, part of the whole preneoplasia picture. And there's a whole field that's focused on what's called inflammatory memory. There've been some high-profile papers on that. And so where that inflammatory memory exists, it makes sense that part of it's going to be in the nervous system, right? So.
Yeah. And, you highlight there the incredibly important crosstalk between the nervous system, the immune system, and cancer, which seems to be central to so much of cancer pathophysiology.
Yeah, exactly.
That's really really interesting and really one of the main future, I think, topics of the field. So Tim, just maybe one comment, and we can discuss this, of course. So you really showed nice examples of potential synapses forming between neurons and cancer cells. I think for the kind of sake of clarity of language, it's probably we probably would need to kind of discuss in the field how to call them, because many of us are now looking into that and find, so the neuroscience people call them putative synapses, because so if they're not meeting all criteria of a synapse, it's a potential synapse or a putative synapse. And it seems to be more established in the from a neuroscience field that when you say pseudosynapse, you say, well, this has some features of a synapse, but we have shown and proven we can't call it a true synapse, but we need to call it a pseudosynapse, because this and that, whatever is missing now, or like the immunological synapse or whatever, you can kind of, this has been kind of used for many. So it might be a good idea to prevent confusion also, to agree to say, okay, these are putative synapses, and which means they still need to be finely characterized with electrophysiology, with EM, etc. But maybe we might want to think about avoiding the pseudosynapse terminology, and just use it when we have proven that this is not a true bona fide synapse with all features. But what are your thoughts?
I'm certainly open to whatever the language should be. I've struggled with this, as you can see. But I think, putative or pseudosynapse would be fine. Yeah, absolutely. Yeah, agree.
Thank you.
Okay, thank you.
Wonderful. Thank you so much, Tim, for the wonderful talk.
Pleasure.
So it's my great pleasure now to introduce our next speaker. The next speaker is Yuan Pan. She's an assistant professor in the Department of Symptom Research at the University of Texas MD Anderson Cancer Center. She received her PhD degree from the University of Iowa and postdoctoral training with Dr. David Gutman, or Gutman, as we say in Germany, and Michelle Monje here, and she redid seminal and groundbreaking work on neurofibromatosis type 1, NF1, and cancer neuroscience, so studying the impact of cancer predisposition syndromes, and integrating those into the cancer neuroscience realm, so to say. The previous work has revealed that light-induced neuronal activity synchronizes with germline NF1 mutation, and we will learn about that to drive initiation and maintenance of optic glioma. And now the Penn Lab, after getting independence, she is now working on understanding how cancer-related mutations influence neuron-glial interactions and how the disruption of these processes contributes to nervous system tumors and neurogenetic diseases. In the end, these efforts are aimed to develop potential strategies for predicting, treating, preventing nervous system conditions, and enhancing patients' quality of life. So really looking forward, Jan, to learn about your previous, recent, and future research.
Thank you so much, Frank, for the introduction, and congrats to you and Michelle for this Brain Prize. Let me see if I can share. Can you see the slides fine? Yeah, okay. Yeah, I would like to thank the organizers for this wonderful opportunity for me to share our work in here. So I'm glad to hear Dr. Wang talking about the sensory nervous system, because this is also what I'm going to talk about. So broadly, the sensory nervous system uses specialized receptors to collect information about internal and external environment and then they transform this information into neuronal signals. So for example, right now you are listening to me. So the hair cells in the auditory pathway, they pick up the vibration from the sound waves and they activate in the downstream neurons. So you're looking at the slides, the photoreceptors in the retina turns the light signal into electrical signals among multiple layers of neurons in the optic pathway. And if you have a cup of coffee near you, you feel the temperature, the texture of the cup. You are using the mechanoreceptors and thermoreceptors in your skin. And if you smell the coffee, you taste the coffee, you are using the receptors and neurons in the olfactory and the gustatory pathways. So this is basically how we sense the world. And what's very interesting for cancer neuroscience is that there are many different types of tumors that reside within or near these sensory neuronal pathways. And these are just some examples in here. So raising a really interesting question, which is, if this is how we sense the world, can the tumors do the same? Or in other words, do sensory experiences shape tumor progression? So we became really interested in this question pretty early when I was a postdoc with Michelle and Dr. David Gutman. And we focused on a cancer predisposition syndrome called neurofibromatosis type 1 or NF1 because for this disease, patients tend to develop tumors within the nerves, as both in the CNS and PNS. So in the CNS, it tends to be within the optic pathway, and in the PNS, those can be classiform neurofibroma or malignant peripheral nerve sheath tumors, which I'm going to talk about later. So brief introduction about NF1. It's a genetic disease that affects 1 out of 3,000 people worldwide. Patients are born with a germline mutation in the one functional allele of NF1. And over time, they may acquire second hit NF1 inactivation in some cell types. And if that happens to be a tumor initiating cells, that can predispose them to tumor formation. So in order to study the biology of the optic pathway gliomas associated with NF1, the Gutman lab developed a mouse model where they recapitulate the germline mutation within the patients, as well as the NF1 null condition in the glioma initiating cells. So what you're looking at here are the two optic nerves connecting to the eyes. So you can see that in the optic glioma mice, there is enlarged volume of the tissue and increased proliferation index. And if we look at these two parameters over time, and about 9 weeks of age is when the mice exhibit both increased optic nerve volume and the proliferation index, and that's when we define tumor formation for this mouse model. So looking further into the microenvironment of those tumor cells, it's very intriguing because they are in the optic nerves, so the only neuronal component they can see is the retinal ganglion cells. And those neurons are the long projection neurons connecting the retina to the brain, and their activity is modulated by light. So our first question is light important for pathophysiology of these tumors? Because it's linked to neuronal activity. So we started with a simple experiment, basically dark rearing the mice, and we started dark rearing the mice before tumor formation. And the data depicted here is kind of like a T-SNE plot. Each dot is one mouse, and then we plotted their proliferation index and their tumor volume, and the tumor cohesion is at the upper right corner, and the gray area is the value range for the wild-type equivalent controls, which are the NF1 flox mice. So you can see that all the yellow dots are the optic glioma mice, which were raised in regular 12-hour light, 12-hour dark cycles. They all developed tumors. And then the dark-reared optic glioma mice, the blue squares, they cluster with the wild-type equivalent controls. And if we further look into the values, they didn't form the tumor. And because the gliomas reside within the optic nerve, they damage the retinal ganglion cells. So in the regular optic glioma mouse model, if you look at the yellow column there compared to the wild-type controls, they exhibit reduced number of retinal ganglion cells, the neurons and the retina. But in the dark reared group shown in blue there, as we didn't see any tumor formation, so there is no effect on the retinal ganglion cell numbers in the retina. So that means light is necessary for the formation of the NF1 optic glioma. And because light modulates activity of the neurons within the optic nerve, we then ask whether optic nerve neuronal activity can directly regulate optic glioma pathogenesis. So we use optogenetics in this, where we genetically expressed trendorhodopsin 2 in the retinal ganglion cells, so the neurons within the optic nerves. Then we put an optic fiber on top of the optic nerve to deliver blue light to activate those neurons. We did it for 6 weeks, and then after that, we took the tumor and we saw that neuronal stimulation increased the size of those tumors compared to the sham controls with the same genotypes. And then to understand the molecular mechanisms behind it, we focus on secretory factors because we saw many examples of where neurons will produce paracrine factors to influence tumor pathophysiology. So here we developed this explant system where the eyeball and the optic nerve were taken out, and then the front part of the eye was removed to expose the neuronal layer. And then again, we use optogenetics to induce activity of the neurons. We did this in NF1 heterozygous mice most of the time because we want to recapitulate the germline mutation in the patient. So after neuronal stimulation, we collected the media for proteomics. So we found that activating the neurons can increase secretion of BDNF, which is our positive control. This is expected. And we also found increased level of neuroligin 3. The neuroligin 3 is really interesting because previous work from the Monje lab led by Dr. Hamza Venkatesh showed that the neuronal activity in the brain can increase the shedding of neuroligin 3, which is a membrane protein. And then the shed form of neuroligin 3 serves as a mitogen for high-grade glioma. So this study was in the brain in a different type of glioma. So we went on to look at the eye by injecting tetrodotoxin into the eye to block neuronal activity in the retina and the optic nerve. Then we saw that shed neuroligin 3 level is reduced within the optic nerves. And we also use light physiological condition to modulate neuronal activity. And similarly, we found that light can also increase neuroligin 3 shedding within the optic nerves. However, if we did the same experiment in the wild-type mice, we didn't see this effect. So the same is true if we use our explant system, looking at what's secreted from the retina. And we also found this neuronal activity increased shed neuroligin 3 levels only in the NF1 heterozygous condition, not in the wild-type condition. When we look one layer up of the signaling pathway, looking at the protein that cleaves neuroligin 3, which is ADAM10, which is also released, we also found that neuronal activity can increase ADAM10 secretion. And this is, again, only applicable to the NF1 heterozygous condition. So together, this is showing that NF1 heterozygosity, or the germline mutation, is required for the activity-mediated ADAM10 secretion and neuroligin 3 shedding. So that reminded us this early study from Gottman lab and using a similar mouse model. So basically, if you look at the second row, this is a very similar mouse model, only that the background is wild type, not NF1 heterozygous. But in these mice, they didn't form optic glioma. So indicating that the NF1 heterozygosity is required for optic gliomagenesis. And given these findings, we hypothesized that the NF1 heterozygous microenvironment promotes the secretion of ADAM10 and increase the level of shed neuroligin 3 that drives optic gliomagenesis. So in order to test whether the ADAM10 shed neuroligin 3 axis is required for optic gliomagenesis. We started with a genetic model. So this is where we knock out neuroligin 3 from the optic glioma mice. So if you look at the plot here, the yellow dots are the regular optic glioma mice. The blue dots are the same mice without neuroligin 3. So with neuroligin 3 deletion, we observed fewer and smaller tumors. And we also use pharmacological approach to block ADAM10. And we saw similar phenotype as the neuroligin 3 genetic knockout, meaning that the ADAM10 inhibition and neuroligin 3 knockout inhibits optic gliomagenesis. And we also found that even after the tumor has formed, we can use ADAM10 inhibitor to treat the mice and then reduce, it was able to reduce the volume and proliferation of the tumors. And similarly, if we did drug rearing after the tumor has formed, we also phenocopy what we saw with the ADAM10 inhibition. So together, what we show in the optic glioma mice is that the light or light-induced neuronal activity in the optic pathway is required for the formation and maintenance of NF1 optic gliomas. And this is driven by an ADAM10 neuroligin 3 mediated signaling. And we were very happy to see that a year later, there's a study published by Cheng Liu's group showing that the olfactory experience can drive the initiation or tumorigenesis of high-grade gliomas that form within the olfactory bulb. And they also found this is mediated by neuronal activity of the olfactory pathway through a different molecular mechanism mediated by IGF-1. So our studies together indicate that the sensory experiences can drive the gliomagenesis in the CNS. So which opened up many other questions. So for example, what about the other sensory receptors? The somatosensory system is extremely interesting to me because it has multiple different types of receptors, and some of them can sense temperature, some of them sense mechanical forces, body position, and also the nauseous signals. And before we focus on nerves in the CNS, how about tumors that develop within nerves in the peripheral? So with this question, we started looking at a tumor type called malignant peripheral nerve sheath tumor, or MPNST. So these are soft tissue sarcoma that forms within the peripheral nerves. They have poor prognosis, and because of its location, it can disrupt sensory neuron function. Right now, surgery is the only curative treatment, but not always possible because of sometimes the deep location of the tumors or the metastasis. So highlighting there is an urgent need for novel therapeutic strategies. So in terms of cancer neuroscience, the role of neurons for this type of tumor is unclear. So we formed a team to look into this question. So this is a study led by Khalil, who is a talented postdoc in our group, and is in collaboration with doctors Herbie, Shepard, and Grace. And the way we model MPNST is that we injected the mouse MPNST cells into immunocompetent mice into the sciatic nerves. So the sciatic nerve is a great place for us because it contains many different types of the sensory receptors or neurons. And for example, as I mentioned before, there are thermoreceptors, mechanoreceptors, and each of the sensory neuron subtype, they respond to their corresponding sensory stimulus. So in the sciatic nerve, those sensory neurons innervate the hind paw. So we can apply the stimulus at the hind paw. One is that we can study how the tumors influence sensory neuron function. And then we can also study how those sensory stimuli influence tumor pathogenesis. So we started looking at how tumor cells influence sensory neuron function. And we did this using behavioral assays. And these are also assays that were commonly used by pain researchers. So starting with mechanoreceptors, we look at we use Wang test, which is using non-noxious mechanical stimulus, the thin Wang filament, to poke at the paw of the mouse and see what's the force threshold for them to withdraw their paw. So if you look at the orange line in there, that's the tumor-bearing mice. So compared to the sham controls, they exhibit mechanical allodynia, so they're hypersensitive to the stimulus. And similarly, we did a pinprick test. So this is using noxious stimulus, a pinprick, to apply at the paw, not piercing the skin, and then see how long the mouse reacts to the stimulus. And the tumor-bearing mice also showed pinprick hyperalgesia. So in both cases, the neurons are hypersensitive to the mechanical stimuli. And now we look at the other way of the communication, looking at how sensory neuron activity can influence tumor progression. So we did basically the same assay, and then we just have another group of mice, tumor-bearing without the sensory stimulation. And for mechanoreceptors, actually in neither case, we saw a big change in tumor growth. So we move on to look at thermoreceptors. So looking at thermoreceptors, we did a Hargreaves test, which is to apply a very tiny heat spot, about 40 to 50 degrees Celsius, at the hind paw of the mouse, and then look at how soon they withdraw their paws. And again, the tumor-bearing mice showed thermal hyperalgesia. And reversely, when we compare unstimulated versus the thermostimulated mice, we saw a large increase in tumor growth. So looking into what's contributing to this, and this is going to be the same neuron type as Dr. Wang was talking about. So these are CHIP-1 positive neurons, because CHIP-1 is the calcium channel that's sensitive to heat. So we, for just generally testing, we did the heat stimulation experiment in wild type versus CHIP-1 knockout mice. We saw that can block the tumor growth. We also applied a CHIP-1 antagonist to the mice, and we saw that the antagonist can block the heat-induced tumor growth. And we did a control experiment in vitro, just adding the antagonist to the tumor cells. We didn't see a large direct effect for the CHIP-1 antagonist on tumor cell viability. And then moving forward, we wanted to look at the CHIP-1 sensory neuron activity specifically, rather than just globally deleting CHIP-1 or using an antagonist. So we move on to use this methodology developed by the Wolf Lab before. So they use capsaicin, which is an agonist for CHIP-1, to open up the channel on the neurons, and that allows the entrance of a membrane impermeable sodium channel blocker, QX314. And using this strategy, they can specifically inhibit the activity of the CHIP-1 neurons. So by using this strategy, we were also able to block the heat-induced tumor growth. And as a cancer neuroscientist, we like optogenetics or chemogenetics. So we also developed a chemogenetic system, a little different from the complete genetic models. So here we use the CHIP-1 Cree mice, and those are adult mice. We injected AAVs into the sciatic nerve, and the AAVs carry the DIO and then the inhibitory jet. So upon stimulation of the jet or inhibition of the neurons, we were also able to see the reduction of heat-induced tumor growth. And importantly, without the heat stimulation, when we inhibit the activity of the CHIP-1 neurons, we were also able to reduce the tumor size at baseline. So together, to sum up the stories or data I mentioned in both the CNS and PNS nerves, And together with other data from other group, I hope that forms a picture or concept that the environmental stimuli can feed into the sensory nervous system by activating on the sensory neural activity to drive the pathophysiology of nervous system tumors. And with that, I would like to thank everyone in my group, also members from Monje and Guttman labs that contributed to the optic glioma study, as well as the collaborators that contributed to the data I presented. And last but not least, the generous support from our funding programs. And I'm happy to answer any questions. Thank you.
Thank you so much, Yuan. Beautiful, beautiful talk. And it's so, every time I see the MPNST study, it gets more and more exciting. That is just, really such an important study and such an important tumor type and really a new concept. Just lovely. Let me open things up for questions. Tim.
Yeah, that was really wonderful, Yuan. I learned so much. And so I love the story about all these different sensory nerve signals you went through that could potentially induce these neural tumors. One of my questions is about how early this sort of stimulation or growth stimulation starts. And it's a little bit different, I think, with these neural tumors in that they're already close to the nerves, right? So you don't have to necessarily bring them in. But are the glial cell progenitors that generate these tumors, presumably whether they're astrocyte types or OPCs or whatever the cell of origin is, are they at baseline connected, or to start receiving these sorts of signals, do they need, does knockout of NF gene, for example, sort of jumpstart these connections in some way, or is it just there from the very beginning? And so as soon as you have a combination of a neural signal and the loss of neurofibromatosis gene, that this is what the combination that drives growth?
Yeah, that's a great question. So in the optic glioma, we know we definitely need both the germline mutation, the heterozygosity, as well as the NF1 condition in the precursor cells to form the tumor. So from at this point, the things we have looked at is only from the neuronal side and the NF1 heterozygous background side. So we haven't, in the optic nerve, we haven't really looked at, like early on when we lose both copies of NF1 in the progenitor cells, how that influenced the connection or their crosstalk to the retinal ganglion cells. But we did something in the brain, which is our next study. We look at how NF1 mutant OPCs communicate to neurons in the brain. But those were wild-type neurons in the premotor cortex. So very interestingly, after NF1 knockout from the progenitor cells, they lose their response to the neuronal activity. So it's a little different from the optic nerve as what we would thought. But also thinking about what neuronal activity does to progenitor cells in the healthy wild-type condition, the neuronal activity promotes differentiation of progenitors. But if a progenitor cell want to be a tumor cell or pre-tumor state, they probably don't like this differentiation process, right? So maybe there is a step where they kind of reject neuronal activity dependent signal. And then after a while, when they are ready, which we're still trying to find out what is the ready status for them. And then they would turn into a state that they were hypersensitive to the neuronal activity signals.
Yeah, I like that model. I mean, there's a lot of data on loss of tumor suppressor genes that one of the effects of loss of tumor suppressor genes is it causes a differentiation arrest. So a stimulatory signal that could induce differentiation would then instead induce, for example, symmetric division and expansion of progenitors as opposed to differentiation. So that may be the model. That's really interesting. Thank you so much, Yuan.
Yeah, thank you so much, Yuan. A really wonderful talk and a great development. Every time I see more and more really interesting avenues. So I think it's a great idea to first focus more on nerve tumors to really map the different effects of the various players, so to say, of the sensory and nerves and mechanosensory and thermal, etc. So I think that's really nice. Do you believe these findings can be translated to other cancers? I mean, people have also started to find different effects of various classes of sensory neurons, or even various sub populations of receptors for sensory input to be really specifically relevant for certain cancers. Do you think this is really something that you can expand to a lot of cancer types? Is this rather cancer specific? Can this be also extended to the cancer types beyond cancer predisposition syndromes, etc.? So what is your take there?
Yeah, so just from reading and going to all our cancer neuroscience meetings, there is a feeling that TRPV1 has become really popular. TRPV1 neurons for different types of cancer types. So many groups found the TRPV1 neurons are important driver for tumor progression or migration, as Dr. Wang was showing. So at least it does give us the feeling that the nociceptors are big drivers for cancer progression. It could be a very broad theme. In terms of mechanoreceptors, it's short. There were evidence for some types of cancer or tumor. It's not as broad as the nociceptors or thermoreceptors we are seeing. So I think that we still need more studies on that. And not to mention the proprioceptors, I don't think we know a lot about that. So I guess depending on the cancer types and the neurons we study, there are some that's shared, some that's specific to the neurons and to the tumor types.
When you let's say make a guess, so comparing really local microenvironmental crosstalk and then on a more brain-body cancer communication level, what do you think? What is the key mechanism here, or are both important? So micro versus cancer neuroscience macro environment, so to say.
Yeah. So a lot of studies focus on the neural immune crosstalk. So it's definitely a big contributor because many times neuronal activity gets suppressed immune microenvironment that drives cancer progression, which doesn't really exclude other possibilities because it can be the neurons directly acting on the tumor cells, like the synapses we were talking about. And it can be other cell types that's contributing, which I don't think we look deep enough into those. That's some areas that are still quite open there. Yeah, and I think we should look into that. For example, the fibroblast angle is really fascinating.
And let's say cancer brain circuits that are then kind of driving again tumor progression. I mean, here nociceptor neurons, of course, would make a lot of sense, right? So by relaying pain and perceptions, and then executing, let's say, stress responses or others that are, of course, have potential to drive immunosuppression, but also maybe potentially actively drive cancer growth.
Yeah. We had that discussion a lot for MPNST because the behavior phenotypes we're seeing is basically means they have pain, and that's continuous. And there should be some central mechanisms in there, and maybe rewiring of the central circuitry that's relevant to pain perception, and how that influences tumor progression. We don't know yet, but we're hoping to first of all understand what's changed, and then perturb that pathways to see what the influence is to the tumors.
Wonderful, thank you.
Wonderful. Thank you so much, Yuan. Just fantastic work, and it's so exciting to see what's coming out of your new laboratory. We'll move on now to our third and final speaker, Filippo Bellaggia, who has been doing some just really, really exciting work. Filippo Bellaggia is a junior group leader at the Department of Translational Genomics at University Hospital Cologne. He studied medicine in Bologna and obtained a PhD in human genetics at University of Cologne. The Balaji laboratory focuses on functional cancer genomics and small cell lung cancer, and his research has recently uncovered the ability of small cell lung cancer cells to form functional synapses and engage in neurotransmission with neurons, which has been a really exciting new dimension in cancer neuroscience, underscoring the ability of non-CNS derived tumors to interact in this really classically neuronal way in the central nervous system. So really excited to hear your talk. Thank you so much for joining us, Filippo.
Thank you, Michelle. All right, I hope you can see my screen. Okay, so first of all, I'm really honored to be here to celebrate you, Frank and Michelle, and I'm really grateful for your work because this it has inspired us and guided us as we move the first steps in this field. And Michelle, it was also really a pleasure to coordinate with you so that we could publish the synapses in small cell lung cancer at the same time. I will talk today about the surprising finding that small cell lung cancers can form synapses. So a peripheral tumor can actually engage in neurotransmission with neurons. Small cell lung cancer affects about 300,000 people per year. It is a very aggressive disease that has usually already spread to other organs before diagnosis. The therapeutic options are limited. Usually, this is a platinum agent and etoposide, so chemotherapy, and more recently, together with immunotherapy. And usually, the patients respond, but then quickly relapse, leading to a median survival of only about 1 year. And therefore, there is really an urgent need for new therapeutic options and for finding new avenues of research. A striking finding at the genetic level is that really the vast majority, potentially all patients, show B-allelic loss of RB1 and P53. And we therefore use alleles, conditional knockout alleles of these two genes in our mice to replicate small cell lung cancer. This is a very well established model that always results in small cell lung cancer. But we are interested in finding new therapeutic targets. And so we crossed a screening system into this model composed of two elements: transposons, many copies of the transposons, and a piggyback transposase, which can mobilize the transposons and reintegrate them randomly across the genome. If the transposons integrate within a gene, then they can stop its expression, simulating the loss of a tumor suppressor. If they integrate outside of the gene, they have a strong promoter which can increase their expression, simulating oncogenic activation, and it has been shown even generating novel oncogenic isoforms. So we have a bidirectional screening system to identify both tumor suppressors and oncogenes. And so these are the alleles. We have RB1, P53, the transposons, and the transposase. And we actually have two lines. In the first, RPLH, the transposons are initially on chromosome 5, and in the other line, RPLS, they are initially on chromosome 10. These mice really generate small cell lung cancer. So these are cells with small cells with very little cytoplasm that express neuroendocrine markers such as NCAM1 and synaptophysin. And we sequence more than 300 tumors and try to identify genes in which there is an increased amount of transposon insertions than would be expected by chance. We did identify some genes that are known in small cell lung cancer, such as CRABP, NFib, and P73, showing that the system at least potentially can work very well. But all of the other genes were not known to us, and they were all involved in, or almost all involved in, neuronal functions and structures such as the synapse. And importantly, they were really identified from both lines. So both starting from chromosome 5 and from chromosome 10, the transposons end up in those neuronal genes. And initially, we thought this may actually be a mistake or just a side effect of the neuronal differentiation of this, or neuroendocrine differentiation of these cells, because it is known that the transposition is more efficient in highly expressed genes. And so we looked for a way to filter out this data and turn to human samples. So we collected genetic mutations from the literature. So these are mutations present in the cancer, but not in the healthy cells. We reanalyzed them. And indeed, the background is really opposite. So while our insertions in the mice tend to be more common in highly expressed genes, the mutations are more common in genes that are very lowly expressed. So this we thought was a really nice filter, but actually it increased the enrichment of neuronal and synaptic genes. For example, you see here neurexin-1, so a synaptic adhesion protein. On the left, mutation in human samples in the lollipop plot, and in the right, the insertions in our mice. So very clear, significant enrichment. And the same, just for example, on neuroligin-1, which is an interaction pattern of neurexin on the synapse. And more in general, the gene sets that were enriched at the genetic level, both in our mice and in human patients, all had something to do with neurons. The top scorer was the synaptic membrane, and we were also quite interested in the glutamatergic synapse because it seemed somewhat specific. However, before embarking in validation, we wanted to make sure that these gene sets are actually expressed in small cell lung cancer. And so we collected from the literature expression data from human samples and compared it to healthy tissues from the GTX database and to other cancers from the Cancer Genome Atlas. The gene sets that were highly and specifically expressed in small cell lung cancer tend to group in two themes. First, the proliferation, cell proliferation, which I think is not surprising because, as I mentioned, RB1 is always mutated, almost always mutated in these patients. And RB1 is a master regulator of the cell cycle. But then we also saw the neuronal gene sets, which in many cases were really the very same gene sets that were mutated in human samples and inserted in our mice, such as the synaptic membrane and the glutamatergic synapse. And this again included individual genes, just as an example, such as neurexin-1, which you see on the left, highly expressed in individual samples SCLC, as well as in general in SCLC compared to other types of cancer or healthy tissues. And neuroligin, for which small cell lung cancer is also one of the most highly expressed in cancers. And as a final proof that this was really something interesting, we look at single nuclei RNA-seq data in our mice, and here we can clearly distinguish the tumor cells from the other healthy cells in the lung based on the expression of neuroendocrine markers. And if we compare the tumor cells here in blue to the other cell types, again we see these two themes, proliferation and neuronal gene sets such as synaptic membrane and glutamatergic synapse. And so at this point, we have both genetic data in human and mice and expression data in human and mice, all pointing to the synapses being an important feature of small cell lung cancer. And we decided to start looking at them. We detected different types of fibers near the tumors in our mice. For example, here you see substance P-positive fibers, which do not penetrate, or at least we haven't seen them, to penetrate the tumors. They're probably from the dorsal root ganglia. P2X3 positive fibers, probably vagal sensory fibers, which do sometimes penetrate the tumors, as you see here. And VGLUT1 positive fibers, again, sensory vagal fibers most likely, which are already present when the first cells start to transform, here in blue, in some of the small tumors, sometimes when the tumors also lose tissue architecture, but we didn't really see them in large tumors. So probably these fibers are important, at least in the lung for this type of cancer at the initial phases. And since we are looking for synapses, we decided to zoom in on the contacts between the cancer cells here in white and the nerve fibers in red using super resolution microscopy. And here we stained for VGLUT1, so a vesicular transporter that loads glutamate onto synaptic vesicle, and HOMER1 as a postsynaptic marker. And so we have a presynaptic and a postsynaptic marker, and we saw a very striking co-localization of these two markers on the cancer cells in the lung of our autochthonous mice. We could reproduce this in vitro by culturing the cancer cells together with the nodose ganglia of the vagus nerve. And here again, the white cancer cells are contacted by the red axons of the nodose ganglia. And again, VGLUT1 as a presynaptic marker, so in yellow, and HOMER1 as a postsynaptic marker co-localize on the cancer cells. We see similar contacts if we transplant the cancer cells in the brain of recipient mice. And here we see that the green neurons dock onto the red cancer cells with what appear to be glutamatergic boutons, because they're marked again by VGLUT1. And then back to in vitro setting, in co-culture with cortical neurons, we used a combination of super resolution and expansion microscopy to really resolve that the presynaptic VGLUT1 positive boutons are outside of the cancer cells, whereas the HOMER1 postsynaptic sites are within the cancer cells. And with this technology, we could also measure the distance between the presynaptic vesicles and the postsynaptic structures in neuron to cancer synapses. And we saw that this distance is really not distinguishable from that in neuron to neuron synapses, suggesting that these are structurally quite similar. And finally, we zoomed in even more detail using correlative light electron microscopy. And this allows us to identify the tumor mass as a red fluorescent mass, and then individual red fluorescent cancer cells, and then zoom in into non-fluorescent neuronal boutons that contact the cancer cells to resolve their ultrastructures. And we do see the synaptic clefts and vesicles that are very close to the membrane, potentially on the way to fusion. This is another example. And the quantification. And we didn't quantify the whole volume of the cancer cells, but just the slices or perimeters. And at the periphery of the transplanted lesions, we see around 8% of the cancer cells having at least, of the cancer perimeters, having at least one synaptic contacts with a neuron. We saw the same in vitro in our co-cultures, and we decided to then use this system to dig a little deeper into the functionality of these synapses. And the first experiment that we performed is synaptic tracing with rabies virus. We wanted to know whether the synapses are able to transmit rabies virus like real synapses do. And in this setup, only the cancer cells can initially uptake the virus because they express the TVA receptor, and only the cancer cells can transmit the virus to a presynaptic cell because they express the glycoprotein G. So this is a monosynaptic tracing that can only start on the cancer cells. And initially we see the double positive cancer cells, initially red, becoming green once they're infected. And then after 1 or 2 days, the neurons start lighting up, indicating that they are connected. And we see a high level of infection of the neurons only if the cancer cells really express both the receptor and the glycoprotein. We see a lot less, probably unspecific uptake if they do not express all components. On average, on 2 different cancer lines, we see 3 to 12 neurons synaptically connected to each cancer cell. We then repeated this experiment in vivo by injecting the cancer cells together with rabies. And again, only the cancer cells can uptake and deliver, only the cancer cells can uptake and deliver the rabies virus. And also here we see the neurons around the transplanted area lighting up, both in the hippocampus where we injected the tumor cells and in the nearby regions such as the subiculum. And I'm not an expert, but I am told that from the morphology, these are mainly excitatory neurons, but there is also a smaller component of inhibitory neurons becoming green, so being connected to the cancer cells. If we perform patch clamp measurements on these cancer cells, we see that about 1/3 do have spontaneous postsynaptic currents, indicating that there may be actual neurotransmission. And we then went back to the in vitro system to investigate this electrophysiology in more detail using a human cancer cell line. So in the monoculture that you see here, we did not see any currents, no matter at what voltage we measured. However, in the co-cultures, we still didn't see currents at -70 millivolts, so there is no AMPA-mediated currents in this cell line, but we did see some currents at 0 millivolts, indicating the presence of GABA-A receptor-mediated currents. And most importantly, we saw a lot of currents at +40 millivolts, voltage at which the NMDA receptors can be activated. And we could inhibit these currents using the DAP5, which is a very specific inhibitor of NMDA receptors. Some currents remain, which is likely the same GABA currents that we saw before, and these we can inhibit then with gabazine, which inhibits the GABA-A receptors. If we wash out the inhibitors, then the currents come back. And on the right is a quantification. So in all cells, we could completely abolish the currents using NMDA and or GABA receptors, inhibitors. But to actually prove that there is direct synaptic transmission, we turn to optogenetic system. And here the neurons express channel rhodopsin, so they can be activated by light, and we measure the currents on the cancer cells. Indeed, as soon as we pulse the neurons with blue light, the cancer cells receive the synaptic inputs and show the postsynaptic currents, really proving that there is direct synaptic transmission. For the most part, these were inhibited by DAP5, meaning again that they are NMDA mediated. And in one cell, the DAP5 could only inhibit a small fraction of the total amplitude, and we needed gabazine to then completely inhibit the currents, indicating that these were GABA currents. And this is a quantification, so there is a very significant difference in the amplitude of the currents when using NMDA blockers. We also noticed that the cancer cells proliferate much faster in the presence of the neurons. And this proliferation was in part dependent on neuronal activity, because if we use tetrodotoxin, we can partially rescue this increased proliferation. Similarly, if we compare co-cultures to the monocultures or to the cultures which are exposed to media that is conditioned by neurons, we see that the actual presence of the neurons in the co-culture is important for the full proliferation advantage. If we only use conditioned media, the advantage is either non-existent, potentially even detrimental, but always at least clearly reduced. And this is true for both highly neuroendocrine subtypes of small cell lung cancer expressed in ASHL1 and NeuroD1, or the less neuroendocrine subtype expressed in POT2F3. We saw a similar phenotype when culturing the cancer cells with peripheral neurons, and these are the nodose ganglia of the vagus nerve, where you can see that the cancer cells proliferate much faster in the presence of the ganglia than without them. And again, this is true for all three different, well-characterized different subtypes of small cell lung cancer. And finally, we started to think about how to target these systems therapeutically, and we decided to try and activate GRM8 which is an inhibitory G protein-coupled receptor that is activated usually by glutamate and is highly expressed in small cell lung cancer samples, both of the highly neuroendocrine and the less neuroendocrine subtypes. In human data from the literature, this receptor is only expressed in small cell lung cancer cells in the lung, and the same goes for our mice. Most importantly, in human mutation data, we see that there is an enrichment in loss of function mutations in GRM8. So we think that the tumor has an advantage if GRM8 is lost, and that is why we try to activate it with a very specific agonist, DCPG. And then as a second therapeutic intervention, we use riluzole, which is an approved inhibitor of glutamate release that is used for amyotrophic lateral sclerosis. So this is how the tumor grows after 4 weeks without treatment. DCPG could clearly slow tumor growth, and the same was true for riluzole. And this is a quantification. This did result in a significantly improved survival for the mice using both agents as single agents. We therefore tested them also in combination with chemotherapy. And here now it's 2 months, so a longer time, because chemotherapy, of course, slows tumor growth. DCPG did not really have an effect on tumor growth, but riluzole clearly and also for quite some time was able to slow or even stop tumor growth. This is a quantification of the response when adding DCPG or riluzole on top of chemotherapy. And for DCPG, this only resulted in a trend towards improved survival, but for riluzole, this was a significant and also quite substantial improvement in survival of the mice. So in summary, we show that neuronal pathways are really a key feature of small cell lung cancer in mouse and human genetic data and in mouse and human expression data. We show that small cell lung cancer can be innervated, that there are many different types of fibers around the tumors. Some of them penetrate into the tumors, especially sensory fibers, VGLUT1 and P2X positive fibers from the vagus nerve. And we saw that there are glutamatergic and GABAergic contacts in the transplantation when we transplant the cells in the brain of the mice. Then we show that the cancer cells can form functional synapses with neurons that can transmit rabies virus, and most importantly, electrical inputs mediated by NMDA and GABA-A receptors. And finally, we show that neuromodulatory drugs can be effective in small cell lung cancer, both as single agents and in combination with chemotherapy. I'm presenting today, but really this was a collaboration that was performed as a unit from 6 different laboratories, including Christian Reinhardt, Lisa Motori, Matteo Bergami, Silvio Rizzoli, and Max Anstotz. In red, you see the people who actually did the work, and all the other people who also participated and made this possible. We'd also like to thank the funding agency for their generous funding, and you and the people watching for your attention.
Wonderful talk. Just beautiful work and such important clinical implications. I think maybe I know there are lots of questions on the panel, but along those lines, the really is all findings are especially exciting given how clinically devastating small cell lung cancer, brain metastases, and small cell lung cancer itself are. Are you aware of any plans to translate this to a clinical trial?
Yes, we do have plans. It has not started yet, but we hope to start something next year. Yeah.
Wonderful. I mean, this is one of the really incredibly exciting things about cancer neuroscience that we're, kind of discovering in the context of cancer targets for which there are existing, neuroactive, medications, and repurposing them in this context can really be transformative. So I'm so excited to hear about the clinical trial plans.
Yeah, I agree. I think repurposing is really one of the most important goals for the next years.
Tim, you have a question.
Yes, thank you. That was really a wonderful talk. I learned so much, Filippo. It was a complicated study, and I think you're explaining it really helped me understand it, than having just read it. So I asked this question before, but how early do you think these sorts of synapses are established? I mean, I guess coming back to the cell of origin of small cell lung cancer, is it a basal cell now that people think is cell of origin? Are the synapses there at the beginning? And do you think it's just glutamate, or could there be other signals from nerves that drive cancer growth as well?
Yeah, so if you had asked me 2 months ago, I would have said this is the pulmonary neuroendocrine cells and the synapses are there from the beginning. But I guess now I find the studies on the basal cells to be quite convincing. And I think there is going to be at least a large portion of tumors that derive from the basal cells. I would be surprised if they have synapses from the beginning. But the mutations are there from the beginning. So the mutations are really very early in small cell lung cancer. They're almost all clonal due to the many years of smoking. And so this, let's say, modulation of these pathways, I think is a very early event. And so I guess the synapses will then happen fairly quickly at the beginning.
Yeah. Maybe I can add one other line of evidence in the study that Hamza Venkatesh and I led together with Sal Savchuk as the first author. We had looked at yet another neuron type, at the vagus nerve innervation of the lung and tumor initiation. So not getting at whether there are synapses in the primary lung. We only studied synapses in the brain with small cell lung cancer, but we did find that transection of the vagus nerve prior to or at the time of initiation of the tumor actually really prevented tumorigenesis in the lung. So there's some critical role for innervation, parasympathetic innervation of the lung in very early stages. Whether that's synaptic or not is not yet clear.
One other quick question. I mean, you showed the synergy in terms of blocking glutamate receptors along with cisplatin. And I think this question, you probably heard it before, but do you think some of the cisplatin is known to cause neuropathies? Do you think that some of the activity of cisplatin could be by affecting neural innervation to some extent? Is there a way to look at that?
Yes. I think that's quite possible, and it could be quantified, right? So we haven't done that, but I guess that's a very interesting experiment that should be done. I just realized I didn't answer your second question from before, Timothy. So absolutely, I think there's other signals that could be exchanged. Of course, CGRP. We saw GABA genes also popping up in our screen, and a lot of the machinery is the same. And of course, we don't really know if this, so in the lung, we have this co-localization of the pre and post-synaptic markers, but we haven't really resolved the ultrastructure. So we don't know to what extent is a really synapses, whether there is neurotransmission, whether neuropeptides may be another target. Yeah.
Thanks.
Can I ask a question?
Yeah.
Yes, thank you. That's a great talk. So I'm interested in the retrograde viral chasing in the brain. So did you see a wide connection for neurons in other parts of the brain? Is it a local connection? And how do you think the tumor cells will affect the function of those neurons and then like brain function in general?
Yeah, so the system is only monosynaptic. So we cannot really see any circuits or any long-range effects. We only saw local neurons or in areas nearby. I imagine that they will activate the neurons, right? That they will stimulate them, and that I think will affect brain function. I guess Michelle and Hamza also showed that very beautifully in their paper. Yeah.
Yeah, I mean, we found we didn't do rabies monosynaptic tracing, which of course can trace to brain-wide inputs to the tumor, but only across one synapse. We did do electrophysiology that highlighted both GABAergic and glutamatergic synapses, just as Filippo also found. And in our hands, optogenetic stimulation of either glutamatergic or GABAergic neurons drove small cell lung cancer growth in the brain, suggesting a really important functional role for both. But we didn't, query whether there may also be cholinergic or serotonergic or noradrenergic or dopaminergic inputs. I mean, there certainly may be. And I don't know, Filippo, if your gene expression data suggests that there may be a wider repertoire of neurotransmitter receptors.
Absolutely.
Yeah.
And also cancer cell lines, they express all types of neurotransmitter receptors, neuropeptide receptors. But Michelle, you did see increased neuronal activity.
Oh, yes, we did. Yeah. So we saw hyperexcitability induced by the small cell lung cancer cells. But how that influences overall circuit function and cognition needs to be studied in vivo and in future studies. But like glioma, probably, brain metastases like small cell lung cancer, I would hypothesize, induce, brain-wide circuit dysfunction important for cognition and other brain functions. We saw the same kind of hyperexcitability induced by small cell lung cancer on neurons in vitro that we saw, gliomas do. Yeah, lots more to learn.
Yeah. Great, thank you so much. So I think it's now my question will probably lead to the more general discussion because I think it's a question for both Filippo and for Timothy. So now we are trying to understand which of these principles and discoveries from, let's say, brain cancer neuroscience can be transferred to the extracranial cancers, where the mechanisms of neurocancer interactions are still not as well probably developed and described and understood like in the brain. So now both of you, Filippo and Tim, you kind of having studied putative synapses or synaptic structures extracranially and even true or bona fide synapses extracranially, and Filippo, comparing them with your intracranial findings. What is your take on that? Do you believe that in the end you can, because I mean you did your glutamate receptor hit screen, then in extracranial cancers you could nicely see it, and then in CNS neuron cancer interactions with small cell lung cancer in brain metastasis models, etc. So do you believe in the end it's kind of very similar, or do you see distinct differences between neuron cancer interaction mechanisms in the CNS and in the periphery outside the CNS with the peripheral nervous system? Maybe you can comment on this.
Should I start?
Yeah, go ahead, Filippo.
So I think there are differences in the sense that there's a lot more neurons in the brain, and so that is really one of the major components of the microenvironment. Whereas in the periphery, they're very important, I think, but I guess the tumors also have to survive and thrive without them to some extent. So I don't think that they are completely dependent only on the neurons. Or to the same extent that they are in the brain. But at least for small cell lung cancer, the brain is also extremely important, because brain metastases are really a major prognostic factor, and we need a way to treat them. So I think there's definitely going to be a strong effect also in the periphery, but maybe not as pronounced as in the central nervous system, with the exception that the immune system is also going to be affected. And I think that is really going to be a major component. Yeah.
It's remarkable that your talk hits really in this extracranial screen, right? And the primary lung cancer screen, where glutamatergic, GABAergic signaling, right, which you would not kind of normally, broadly not expect to be of such a huge influence in extracranial PNS signaling, right?
Yeah,
this
no,
is right,
but I
of course,
think,
so this could of course speak for kind of an enrichment of also of pathways and of neurotransmitter receptor systems, and that are particularly relevant for cancer, but potentially not so relevant for other physiological functions of the peripheral nervous system outside the brain, right? Is that possible, or what do you think about that?
Sorry, I didn't get the last part of your question.
The fact that like glutamatergic GABAergic signaling kind of really was where he hits in your screen, in your genetic mouse screen, it kind of could argue for that in maybe contrast to a potentially lesser relevance of these pathways for normal PNS function. It could be particularly relevant for PNS cancer interactions, right?
Yeah, so I think they are critical. But as I mentioned, for the initiation, I have the impression that the tumors kind of have to activate these pathways also themselves in the periphery. And I think small cell lung cancer does that, especially if it comes from a basal cell of origin, right? And then completely goes into this neuronal differentiation. And I think that is going to be more relevant maybe in the periphery. But we do see the hits, and I think they're critical. And I mean, Michelle and Hamza showed with the vagotomy that the influence on tumor formation is really extreme.
Yeah, so I could just comment on a couple of points questions that Frank raised. First, so I agree with Filippo completely that the brain tumor microenvironment and the microenvironment for solid tumors is different in the sense of the overall density of neurons is much less. And so, but I don't think that necessarily means that nerves are much less important as a component of the tumor microenvironment. It's not all about numbers per se. And so I guess one of the points that I've been trying to raise is that the connection by sensory nerves and other types of nerves in the microenvironment is extensive. And so I do think that this cancer sensory nerve interaction is sort of critical in driving the total activation of the peripheral nervous system in modulating the tumor microenvironment. And so we're seeing sort of dramatic changes in the tumor microenvironment if you sort of turn off that connection. So I think in the end, it's going to be just as important, but different in that the direct interactions are going to be much less because of the density of nerves. And I guess one of the questions that comes up then is if a small subset of cancer cells are innervated by tumors, by the nervous system, what is the difference between those cancer cells that are innervated and those that maybe are not directly innervated? I mean, they can still be stimulated to grow based on the microenvironment changes. And is it the case that the cells that actually metastasize are primarily the ones that are innervated, which is a hypothesis that's interesting, but really hard to test. So, and I guess the question I'd have for Filippo and others is this glutamate signaling and GABAergic. I mean, I guess the pancreatic cancer paper by Aiken Demir also suggested that glutamate signaling was important, at least for those cancer cells that showed perineural invasion. So do you think that this is a common signaling mechanism for many solid tumors? I mean, we have not investigated this yet in gastric cancer, but it's certainly raised the question as to whether we're missing it, and then what is the overlap for these different signaling molecules between CGRP and glutamate? Are they all active at the same time, or some active in some tumors or not others? I'm trying to wrap my head around this and come up with a way to look at this, but I think it's really interesting.
I agree with you. We haven't looked, to be honest, at other types of cancer. I know that glutamate receptors are expressed in many different types of cancers, as well as on the immune cells. And so I do think glutamate is going to be a key player, along as certainly together with the neuropeptides that you have been working on. But I haven't looked at it personally, so I can only speculate.
Yeah, a couple of points. Maybe sorry, Yuan, go ahead.
Oh, sorry. I just want to add a little bit. So we did a preliminary test where for the MPNST cells, we add glutamate, CGRP, substance P, just to see direct effect. We only saw it with glutamate. So like maybe like you said, it's a general mechanism, but still something we need to look further into.
Yeah. A couple of points. One, we're in the midst of a pan-cancer computational analysis across tumor types, and it's highlighting common principles of neuroscience-related gene expression across all malignancies, as well as tumor-specific biology. And that principle is played out, in brain tumors as a paradigm. We see glutamatergic neurotransmission in gliomas important across, every glioma subtype study. There really are very distinct glioma subtypes, and they have different biology, but they all respond to glutamatergic signaling, and they all seem to express AMPA-dependent synapses. However, in GABAergic synapses, that's a little bit more, tumor glioma subtype specific, where we see really strong evidence of growth promoting GABAergic synapses in diffuse midline gliomas. It may be more heterogeneous or less enriched in IDH wild type glioblastoma. So, we are, clearly identifying some overarching principles of tumor subtype specific biology, and then some common principles that extend across cancers. And I think as the field advances, we'll have a lot more granular detail about how to think about that. One point that I think is really important to make, maybe as we transition into the broader discussion, is that the same principle in the central nervous system is playing out in the peripheral nervous system, which is that the activity of the nervous system, of course, is so important for brain development and plasticity, throughout life. But the same is true in other organs, that peripheral nerves are crucial in innervating tissue stem cell niches required for organogenesis, during development, and then later required for homeostasis and regeneration. And it's those stem cells in various tissues, various organs, that tend to give rise to cancers. And so it makes a lot of sense that the cancers would take advantage of that biology and be regulated by innervation. It's just less appreciated, I think, as we, think about the physiology of non-CNS tissues, that innervation is so important, but it really is. And so, this all makes perfect sense from the perspective of neuroscience, that peripheral nerves would be regulating everything. So, maybe on that topic, we can open things up to a broader discussion.
Yeah, this is certainly what we also see. So in brain tumors, but also in extracranial cancers, metastatic cancers, it's clear that it's the stem or stem-like cell subpopulation that is particularly willing to interact with neurons also. And so I think it's now really and also a population that appears to be able to then develop neuronal expression and other physiological neuronal features. So we have a cancer cell intrinsic neural program that we also already discussed, and I think these cells have some stem-like features, at least in the beginning, and then they are also the prime interaction partners. And the question is, where is egg and hen, etc. But probably it's a bidirectional interaction of these stem cells, normally influenced by neurons and kind of hijacking these physiological influences from neural input on them, and then being driven then and developing into cancer. And it's really striking that so many neural, and this is beyond, of course, beyond small cell lung cancer, which is of course a neuroendocrine cancer type, but so many cancers, bad cancers, bad subpopulations, recurrent tumors, resistant situations in the clinic. There is it seems to be often in pancreatic cancer, in prostate cancer, also probably in lung cancer, breast cancer. There seems to be an increase of neural-like expression, at least patterns, and this is really exciting. I think this is something that we also really should try to understand, how certain cancer cell subpopulations in the same tumor are particularly interactors with the nervous system.
Yeah, I totally agree.
I mean, we do have the means now, right? So there's so many single cell data available, and more can be generated. So I think heterogeneity in the sense of more or less neuronal features driving this interaction thing certainly can be investigated now.
Yeah. And importantly, we also have now the neuroscience tools, which, the talks today have really highlighted to specifically control peripheral nerves, very, very precisely control genetically, defined axon subtypes. We can optogenetically play in, physiological patterns of activity and see how that influences the cancer. We can use optogenetic tools to depolarize the cancer cells and see how that influences the nervous system, as Tim showed so beautifully. I mean, I think there's a lot of potential in modern neuroscience tools to modulate this and ask really important questions about function and pathophysiological relevance. And so, lots to do, particularly as tools in studying peripheral nerves continue to expand and advance.
Yeah, in terms of translating this into the clinic, one of the things that came up in some conversations is, as you know, I mean, for lung cancer, particularly the vagus nerve seems to play a big role, but there are these electrical tools for modulating vagal nerve activity that have been used, in patients with epilepsy and so on. And do you think any of those tools, for those of you that in the neuro field, could be adapted for cancer cell treatments in some ways, or are they too sort of blunt and just a thought?
No. I think functional neurosurgical tools modulating the activity of both the peripheral nervous system and the central nervous system might play a really important role in cancer. And that's something that, it's clear for brain tumors that, functional neurosurgical techniques to modulate malignant circuits, the way that other pathogenic circuits are modulated, might represents a potential direction to go. And I think the same is true for peripheral nervous system interactions with cancer. Lots of work to do to optimize that, and likely those devices or approaches will need to be responsive in their nature. They'll need to adapt to the changes induced by the evolution of the tumor and the evolution of the malignant circuitry. But I think it's a really important therapeutic direction to go.
Yeah, I agree. Actually, we're starting that. We're starting that soon to try vagus nerve stimulation in our mice. So
Great.
I'm very much looking forward to that.
Yeah, great. Wonderful.
Yeah. So do I. Yeah, we have a nice consortium now in Germany where we are collaborating on these points. Yeah.
Wonderful. Well, this has just been such a wonderful and very stimulating session. There's so much to do in the peripheral nervous system. And, one of the big questions, we can only modulate the peripheral nervous system as well as we understand it. And actually, the central nervous system tends to be a bit more studied in neuroscience than the peripheral nervous system.
Yes.
And, so advances in peripheral nervous system neuroscience is really going to advance cancer biology. And one of the kind of, stunning things we don't know well in every organ is, kind of the extensive detailed anatomy of the peripheral nervous system. How do peripheral nerves innervate organs? What's the pattern? What's the branching pattern? Where, that's pretty important to know. So, really detailed peripheral nerve anatomy studies really are still somewhat lacking. Of course, we know where the big branches go, but there's a lot to learn. And there's some people like Alison Lloyd doing some incredibly beautiful work in this area, and I'm just very excited to see that develop, because that fundamental neuroscience is going to really enrich cancer biology.
Yeah.
I think cancer neuroscience may end up helping actually peripheral neuroscience, right? Once the cancer has been traced, then maybe we will understand what is innervating what.
Yeah. Right. And of course, any, cancer innervation kind of has to be done in parallel with the normal tissue innervation to understand how it's changed by the cancer. So, again, peripheral nerve, neuroscientists working in cancer are needed, and cancer biologists collaborating with peripheral nerve neuroscientists is needed. And hopefully this will help energize that aspect of the field.
pursuing a career in neuroscience or neurology or whatever, neuromedicine in the last 20, 30 years, something you were not doing, we're kind of focusing on the peripheral nervous system. I think this, so Filippo, I completely agree, so, and Michelle, so cancer neuroscience can be a great stimulus to change that, because we have not understood everything. It's amazing how little we know about the peripheral nervous system, about the basic anatomy, about the neurotransmitters, about the physiology. It's really amazing. And yeah, now that we are kind of, and we are doing this in Heidelberg, and I mean, so all of you doing that, right? This tracing now, monosynaptic, polysynaptic, on neuron proximity tracing methods that we can really now identify the innervating neurons and profile them and characterize them and understand the circuits in the normal organs, and then in cancer, as Michelle said. So comparing that, I think we will learn a whole lot about what's going on in extracranial cancers, and we will also learn, I think, a lot about what are the best targets, therapeutical targets that really are different between malignant and physiological, and guide us also in selection of the best neuroactive drugs.
Yeah. So I think in the GI tract, there's a lot of interest in the peripheral nerves in terms of sensing, particularly nutrients and other signals, and signals from the gut microbiome, for example. And so there's investigators here who are looking at vagal sensory afferents in terms of nutrient sensing and how that affects appetite, and obviously things like obesity. I think Charles Zucker has been looking at sensing in the intestine in terms of microbial and other nutrients, microbial metabolites, and one of the things to think about in terms of cancer is how the sensory innervation changes during pre-neoplasia. So, for example, I mentioned that chief cells have rampant and are innervated by sensory afferents, but in pre-neoplasia, chief cells are lost in terms of this atrophy metaplasia sequence. And actually, the loss of chief cells actually turns out to rewire the sensory nervous system in ways that can probably drive cancer development. So I think, as you said, we just, in terms of the roles of these sensory nerves in all parts of physiology in the gut, in the skin, the other question is, of course, is, TRPV1 is clearly a pain sensor, but in the gut, there's lots of TRPV1 nerves, but there's not much pain coming from the gut usually. And so how do these different sensory nerves play a role sensing specifically environments of the particular organ that they're innervating? And you can think about the lung and the things that the sensory afferents would be looking for potentially, microbes and other things. So I think the role, for example, of microbes in the lung and in lung cancer is sort of actually an interesting area that several people are working on and looking at right now. So I think, I think as you said, cancer research may drive some of these basic questions about the physiology, normal physiology of that organ.
I think the area you mentioned about studying pre-neoplastic state is super valuable, because we're just the healthy state in a neoplastic state, and it's very understudied for any type of cancer. So if we understand a little bit about that stage, that will make it easier for us to understand both ends.
Yeah.
Yeah, absolutely.
Excellent.
So we need more neuroscience in cancer biology and more cancer biology and neuroscience. We really need these two fields to intersect.
I think the work of a lot of investigators on this webinar has inspired interest. And as I've gone around to I've recently visited Washington University, University of Pennsylvania earlier this week. Everyone now is excited about this field, and everyone is getting into it. So you're going to have a lot of company working in this area.
That's what we want.
That's
what
we
want.
I know that was our goal early on.
And we especially need the neuroscientists to come in.
Right.
I know.
It's a great field for collaboration. I think we have seen this in the past, and we have made great progress via collaboration in the past, and I think this will also determine the future. Yeah, particularly in this really multidisciplinary field. Yeah, without collaboration, we will not make the progress we could.
Yeah, we go further together than alone.
Absolutely.
Wonderful. Well, on that note, this has been just a wonderful discussion, wonderful webinar. Thank you so much, Tim, Yuan, and Filippo. This has just been it's been so much fun this morning.
Thank you
Thank
for
you so much.
organizing this. Thank you so much.
a lot.
It was really an honor to be here.
Thanks to all of you. This was a really wonderful discussion, a lot of fun, and yeah, I would say the third and for now the last edition of our seminar series. But yeah, we hope to see many of you in our Brain Prize Cancer Neuroscience Conference end of August near Copenhagen next year, and this will be an exciting meeting, and we will have so you will see many of the webinar presenters are also presenting news data discussing cancer neuroscience, and yeah, we are looking forward to see many of you there.
Looking forward to it.
Sounds good.
Okay.
Bye.
Bye-bye.